Yeah, hello everyone. Welcome to this Kavli NeuroFutures meeting. My name is Christoph Kirst. I'm an assistant professor at University of California, San Francisco, and also at the Kavli Institute for Fundamental Neuroscience there, as well as a faculty scientist at the Lawrence Berkeley National Lab. I would like to thank the organizers for inviting me here to give the opportunity to talk about some of my work and my computational lab, in particular here with focus on mapping brain activity and structure at cellular resolution. So as neuroscientists, we are of course interested in how the brain is doing computations, enables us to recognize the world, and then ultimately generate intelligent behavior. And that's done via an intricate interplay between these extremely complex brain structure visualized here with one of our clearing tools and imaging. And the complex brain dynamics that you see here in this large-scale recording in the lab of Ali Pasha Balziri at Rockefeller University, where I did my postdoc. And the question is, of course, how does brain structure and complex brain activity interact in order to generate these incredible functions of our brains? So my approach is, as a computational person, is to understand brain computation better as a whole, particularly how can flexible brain function be achieved, and how do different areas in the brain interact in flexible ways to achieve this flexibility. And for that, we need methods to better understand how different brain areas are activated. So we'd like to map the brain as a whole, and particular activity as a whole, as a system. So we like to have tools to do that, to map activity and structures. Those tools, besides helping answering these fundamental questions in neuroscience, would also, of course, help to better understand the effect of diseases on brain states, and also the effects of genetics and pharmacology. People have, of course, tried to record, let's say, brain activity on larger scales, and particularly here, EEG techniques or fMRI techniques enable this to do it with a quite fast time resolution, but a low spatial resolution. You get higher spatial resolution single neurons here by sticking electrodes into the brain, and people do this up to 40 electrodes now, for example, or even more, hundreds. Still, while you get single cell resolution with these techniques, you won't be able to record the full brain. To do that, people have developed optogenetic tools and fast imaging techniques like Misha Arendt's lab here that image, for example, transparent brains, and in this case, the zebrafish larvae, and do calcium imaging in those. And I showed this video before from Ali Pasha's lab, who used new techniques called lattice light beam microscopy that is able to record, in this case, over 1 million neurons from a large fraction of the cortex. However, recording the entire brain activity at cellular resolution has not been achieved so far. So what I would like to introduce here is one method we developed to do that. But of course, while it is very difficult technically to do activity, we completely sacrifice time in some sense in this case. And the way we do it is the following. We make use of the fact that neurons, when they activate and become active, express what people call immediate early genes, and one of those genes is CFOS genes. So by using this fact, we had the idea, if we stain for the CFOS genes and then take the brain, clear it, and image it with, in this case, light sheet microscopy, we will be able to get at least a snapshot of the brain activity, and how the brain was used, let's say, prior to sacrificing the animal and using our protocol. So for this, we had to solve two steps. The first step was to clear brains, and here work by Nico Renier and others in the that started in the Marc Tessy-Lavine's lab at Rockefeller, developed this iDisco method that helps to clear brains. The idea is basically that brain tissue is not transparent because the light gets scattered through the inhomogeneity in refractory index, optical density, so to say, particularly between the lipids and the proteins in the brain. So this method is removing the proteins and makes the brain basically transparent. And here you see the normal brains on the left, and the clear brains you hardly see in this tube because the lipids are removed now by a sequence of solutions applied to the brain. So this technique solved this first step. But then the second step after light sheet imaging imaging, you end up with a huge data set. And this is when Nico approached me asking if I could like help analyzing these data sets as a computational person. And the data was so nice and interesting. We started this project that is still a continuing collaboration, which I'm introducing to you more in detail today. So particularly, I'd like to introduce you some of the computational tools we developed in order to process this data. The challenges of that are, first of all, the data size itself. So in our case, it's terabytes, but higher resolution will yield up to petabytes of data. Bad scaling, the data is 3D. So if you just increase one side by 10, you increase the entire data by a factor of 10 to the 3. That makes algorithms scale very badly. While they run very quick on your small cubes, suddenly you get stuck on your bit data size when you apply it to the whole brain. The nature of the data itself, as you can see visualized by this nice view over here, is quite complex. So extracting interesting structures can be quite challenging as well. On top, you have a microscopy artifact. And then once you have extracted data, you also want to put that data into certain representations of representing the anatomy or other measures in the right way. And these may be huge measures that have to be optimized, and tools have to be developed for that as well. So we try to solve this, in particular, have people install their software on their local workstation to get around like huge cluster computing as well. So the ClearMap pipeline looks as follows. We start with our image stacks that has to be stitched together. While there's a large range of stitching tools, we saw the need to develop our own stitching tool, which we called Wobbly Stitcher. And the reason is the following. If we put together a lot of these stacks, what we see that often those stacks at certain ranges in Z can be nicely put together. But then in other ranges, they are like not overlapping at all. And this cannot be solved by rigid stitching, by moving the stacks. If you move them, then you screw up here and so on. So we saw the need to develop what we call our wobbly stitcher that is able to treat these stacks as some wobbly type of stacks that show these oscillations. And then we can nicely fit our data together. So you're welcome to try this tool if you have similar problems in your data set. This is fully automatic, so you can try that nicely. The next step would be to take the data and, for example, align this to a reference atlas in order to measure the different regions and expression patterns, for example, in different regions. For that, we actually take a second image in the autofluorescent channel. We align that image using sophisticated tools like this elastic in a nonlinear way to the reference that's provided by the Allen Brain Atlas. That also comes with an atlas image that labels all the regions. In this way, we can now label our data set with all the specific regions and do specific measurements that are regional specific, for example. The next step, of course, you want to extract the interesting information. So for that, we developed lots of image processing steps. The first one I already mentioned, you have these shadows like artifacts from the light sheet that come in. So we developed a tool that works on large data very quick. To remove these stripes, you see the result here. The idea basically is we take for each pixel, we calculate some statistics locally in an area. This is aligned with a shadowing artifact of the light sheet. And then we take another area that is a square to measure basically background intensity. If the background intensity is different than what we have here, then that's typically foreground. If it's in a similar range, then that will be a striping artifact, and then we can remove it to get these nice images over here. We have other filtering tools in our pipeline. In the end, after filtering a lot, we will detect cell centers here and also their cell shape, and then measure intensity of the expression, in this case, CFOS expression, which was the activity marker. Once we've done that, we use analysis and group statistics. So because our pipeline is high throughput, we can do this with a large number of samples to generate, for example, these heat maps that tell you like where are lots of cells that express CFOS or where the expression is particularly high, and we can do statistical comparison. Of course, we can take our atlas annotation now and measure in each area the counts of cells that were active, for example. So this solves the second part of our general pipeline that is quite useful. And I just show you one application. There are many applications that you can use this. Here is just one basic study, particular using mapping the effect of drugs in the brain. So what we took here, we took a drug called haloperidol that's used to treat, for example, schizophrenia. Psychoactive drug. We give that to the mouse, let the mouse let it take effect into the mouse. Then we use our clearing pipeline and then analyze our data with ClearMap. And what we find is that there are certain areas that light up, that change due to the application of haloperidol. For example, here, the globus pallidus shows an increase in activity. Similar to the CPU here, these areas have been identified before in being involved in that drug. So this is a good control. But now, because we have the whole brain, we can look at every area individually and check which areas are affected, and maybe we find new areas that haven't been discovered. Well, we see the ones that have been discovered. We see even some that hasn't been discovered that show up here that are not necessarily super statistically significant in this case. But if you look closer, we actually see that a sub-area is only affected, rendering this effect not statistically significant. If you look at these subpixel or at cellular resolution, we make use of our cellular resolution, we now get like a nice sub area in this area that is affected by this drug as known before. But also we find other areas that are affected by this drug. For example, like this primary somatosensory area that is involved in processing information from the mouth, as well as here from the nose. And it's actually known that this haloperidol, if given to humans as a medication, has side effects of dry mouth. So in this case, our tool kind of lights up some areas here that could be used to predict certain side effects. Of course, this tool is not able to explain what's really going on. You would now have to, once these areas show up in your screen, you have to now go and measure maybe with like the electrode recordings introduced before to figure out the precise dynamics that are going on. Okay, so this pipeline worked nice to map activity on the whole brain at cellular resolution, but there's no reason to map other things like brain structure, which is the second project I'd like to introduce you in the remainder of the talk. So instead of mapping for activity like we did in the beginning, particular FOS markers, we now decided to map for vasculature, because the vasculature is also quite a sophisticated anatomical structure in the brain, and also involved in activity measures like, for example, fMRI. The bolt signal is completely reliant on the blood flow and blood oxygenation level. So the structure of the vasculature will have a strong effect on these measures as well. And that was another motivation to go in this direction. For this, we take markers. In this case, we take a combination of markers. In this case, epithelial markers that mark this, basically the vasculature tubes. We apply our protocol over here, IDISCO clearing protocol, and then we do imaging. And in this case, instead of just one marker, we also mark for the smooth muscle cells that are stained by ACTA2, which mark the arteries. So we get like 2 color images in this case, where you can even identify the different vessels as veins versus arteries. And this gives these nice 2 color images. In red here, the arteries, and in green, the veins. Or here, the same, in red arteries, and in blue, the veins in this video. This gives you some idea of our data set we get out of the clearing. You can mark the big vessels that have anatomical names, for example. We can look here in these various areas where we see like the main areas that are fed by main arteries that show even these connections anatomically that were known before. You can look in other areas where the structure is very different. This looks very interesting, like here. For example, here in the cerebellum, you see nice structures and alignment of the vasculature with the various layers in the cerebellum. Here you see, for example, the vasculatures penetrating perpendicular into the brain in the cortical areas. And then this is just a sanity check. Our data is quite isotropic in data due to the fact that we only use a very thin part of our light sheet. Okay, but the initial pipeline, of course, is not going to work to extract these vasculature networks. So we had to rewind a bit and develop some new tools that take this raw data and turn it in the first step into like a binary data telling you what is the vessel and what is the background. This we do by a multi-path binarization, because I told you in the beginning, this data is quite multi-scale. So here, for example, is an example. We have this big vessel that generates like a big halo effect even around, which we can detect and then even get rid of the shadow by what we call pseudo deconvolution. But that's not enough. We have to amplify weaker signals with other pipeline steps, which, for example, here the tubeness filter tries to extract small capillary tubes. And then we have additional adaptive mechanisms to account for intensity changes all across the brain. Once put that together, we get a nice binary image. But remember, I told you that in the beginning, we are marking for the surface of the vasculature tubes. So we get these hollow tubes, unfortunately, which we have to fill. So to do that, we devised like a convolutional neural network, a deep network that is filling these vessels in 3D. But there's, of course, no training data to do that. But nicely, we were able to generate our own training data by, in this case, changing the protocol slightly and not staining for cell walls, but staining for, in this case, actually blood. The challenge there is that the blood can leak out of the vessel. So to avoid that, we're doing this in-skull fixation to keep the blood into the brain, and then we can stain for it. We get filled vessels. We can artificially make them hollow and then train our network with ground truths from that. That works quite nice in our case. So once we have our binary data, we try to extract the structure of the vasculature network. The first thing is to basically extract the center line of these vessels. And to do this, we use a skeletonization algorithm. But if you just go and take a algorithm out of the shelf, you see that on our data, this will, after 1 week, maybe be successful or crash. So this is not an option. So we did a lot of improvements, and this is just one example to speed up this pipeline and make it very efficient. We now have one of the fastest skeletonization algorithms here, I guess, that exists in parallel, that allows you to now skeletonize our data in 40 minutes. So a huge speed up, and quite essential if you want to do high throughput analysis of these data. Once we have our center line, we of course now can extract the shape of these tubes and also the branch points and where these vessels actually branch or join. I told you that we have 2 channels, so we also mark for the arteries. We put this all together and generate this large graph that represents the vasculature network that has labels of artery versus vein marker. And of course, we can now combine it again with our atlas registration and look at the different areas. And just here, a small advertisement for our tools. So besides the tools that I introduced, we have a lot of other tools, for example, tracing tools, etc. in our pipeline. We have a GitHub page and we have a documentation for it. So check out if you like. There's also videos and tutorials on how to use the activity marking and the TubeMap I introduced you now. Once you did this, you get these really beautiful graphs of the brain vasculature, which are quite big and even hard to render by itself. So I scroll through some of those images just for fun, or some video here of some slides. You can fly through a bit to give you an idea how dense this vasculature is. The colors here represent the brain atlas, and red are arteries and blue are veins. Here again, just rendering a small slice and then all the arteries and veins we detected, just to give you some impression. Here you see a bit more detail. You see cortical areas. They seem to have lower density than the subcortical areas in red, and higher density. Of course, can verify that by measuring various, in this case, basic features of the brain vasculature. So this brain of a mouse, which is roughly a centimeter cubed, if you stick together all the vessels, you end up with almost 300 meters of vasculature packed into this small brain. So that's an interesting finding, the first measure of the total length of the vasculature. We can do this and like separate out which brain areas contribute what lengths to it. We can also look at the density, how much vasculature is there per volume, for example. We see this effect I showed before, that in subcortical areas seems to be much higher vasculature in general than in cortical areas. If you zoom even further in, you see even an interesting pattern that reappears here over the different layers in the cortex. You get a systematic variation of the vessel density. And that's similar reflected also, for example, in the number of branch points over here. So it's not just length, it's also branching of the vasculature that is shows similar trends. We can do slightly more sophisticated analysis, like over here, where we, instead of just mentioning the branch point density and length, we also measure, for example, orientation of the vessels and then certain features of where the arteries end and how they branch. If you measure these features over the cortical layers and then calculate the distances, here in this particular case, the Wasserstein distance, and you see two big areas emerging. One that has similar features, and you can actually see that those areas are responsible for sensory processing, and then another big cluster that is mostly motor and integrative sensory processing. So you see these functional properties that are reflected in the vasculature structure of the brain. A final example, we saw that the auditory areas, and here, for example, the auditory cortex had an increased branch density in the mice. So we took a genetically deficient mice that were not able to hear and looked at the density, and we see like a strong decrease in the vasculature density, in this case, in the auditory area. This effect is even more significant in subcortical auditory relays. So here are various nuclei that involve. The one striking example is this cochlear nucleus there, where you lose a lot of vasculature density in the deaf mice, for example. You can look at other areas because we map the whole brain again. And we see interesting effects that are known more on the neural activity side, which are called cross-modal compensation. So because the mice is losing its sense of hearing, it seems to compensate this loss of inputs, sensory inputs, with other sensory inputs, like here, somatosensory inputs. And here in the somatosensory cortical area, you see now an increase in the vasculature density in the deaf mice. So you see this cross-modal compensation even in the vasculature by just looking at the vasculature. And with this, I like to summarize. So today I introduced you into our iDisco ClearMap pipeline that can be used to, in the first part, discover, for example, brain areas that are activated by drugs, but you could also imagine certain types of behavior. We combined that, for example, with also axon tracing. You can see which areas activated, and then how this change in activity is maybe translated to other areas due to axon tracing. There are much more applications of that pipeline. I didn't have the time to go into. Our vasculature pipeline, TubeMap, also has, of course, more applications, for example, to stroke models or to detect other more sophisticated structures and statistics in general of the brain vasculature as a whole. For future directions, we are now scaling up our methods. We like to go to rat brains, and then going beyond structural mappings here, we like to go back and add dynamics. For example, in case of the vasculature, we like to look at like, are there certain specific dynamical patterns that are related to vasculature features. Or I told you fMRI signals are based on blood flow or changes in blood oxygen. How is the vasculature and the changes in those related to the actual structure we can map out here to the capillary level? And then maybe how is flexible brain function supported by flexible energy resources that are provided by the vasculature network? How is the flow mapped by ultrasound, for example, here related to different features in the vasculature? This will cause a lot of data that we need to analyze and also interpret. So this is some of our future directions. With this, I like to particularly, I like to thank you for your attention, first of all, but then I like to thank my long-term collaborator, Nico René. We started both out as postdocs in Marc Tissy-Lavine's lab at Rockefeller. Marc now moved to Stanford. The iDisco project started in close collaboration with Juhao Wu and Eliza Adams. Some of the staining for the vasculature was developed by Alba Prado and Thomas Duplico, and then some of the pipeline development was done by Sophie Scriabin and Paul Bertin. And then I like to say thank my funding sources, of course, the Kavli Institute that helped me actually throughout my career. Before I started at UCSF, I was a Kavli fellow at Rockefeller. And then the ICM, where Nico is now runs his lab, also supported this project quite strongly. With this, I'd like to put one advertisement in, if that's interesting for you. We also do a lot of computational work on the neural activity side. I didn't have the chance to talk about today. We developed tools to better understand large-scale dynamical patterns. So if you're interested, my lab is just starting out. So I have open postdoc and PhD positions. Please don't hesitate to contact me under this email address over here. Our Berkeley lab has a nice view over the Berkeley campus and Bay Area, and then our UCSF part is in this new Weil building in the Mission Bay campus. Yeah, and with this, I'd like to thank you again for your attention and happy to take any questions.